Tonight's Royal Institution discourse is about one of the most serious health hazards to arise in the UK in recent years, the epidemic of mad cow disease, or BSE. The speaker, Professor Roy Anderson, Linacre Professor of Zoology and Director of the Wellcome Trust Centre for Epidemiology of Infectious Disease at the University of Oxford, describes the origins of the crisis and the factors responsible for the epidemic. He shows how lessons must be learnt and how to react to situations with serious possible implications where limited data and analysis are available. This may be an archetypal example of a wide range of situations which are likely to become ever more common in the future.
A few weeks ago, I was asked to give a lecture on the BSE epidemic at Southampton's Oceanographic Institute to an audience of ocean and fisheries scientists. On the train journey from Oxford to London, or to Southampton, I spent some time thinking about the possible connections between the topic of my lecture and indeed the interests of the audience. Now, having failed to think of anything rational to connect the two, I turned to idly flicking through a newspaper of that day and was quite amazed to come across a headline which had the title, "Cow Sinks Chinese Fishing Boat." Well, with my attention totally and utterly captured, I read on to discover that a group of Russian military personnel had stolen some cows near the Chinese border, herded them onto a transport plane, and taken off with the aim of selling them on return to their home base. Now, on the trip, quite understandably, these cows became extremely restless and caused, indeed, a considerable instability in the aircraft, with the result that the crew opened the rear doors of the plane and forced the cattle out of the back. Now, at the time, the aircraft was over the sea and one of these falling cows hit and sank a small Chinese fishing boat. Absolutely true. It became immediately clear to me that the invitation that I'd received was to try and think jointly how to solve two problems: how to dispose of all the cattle that had been culled in an attempt to control the BSE epidemic in the UK, and secondly, how to control overfishing in the North Sea. Now, today I've not been so lucky in finding an obvious link between BSE and the Royal Institution, except that the solution of the BSE crisis requires a very, very clear understanding by politicians, civil servants and the public of the scientific issues that underpin the epidemic and its control. Now, the Royal Institution is clearly the ideal place to attempt such an understanding, and that indeed is my goal tonight. The first case of a mysterious new cow disease was seen by a vet in Ashford, Kent in April 1985. The symptoms of the disease, which were subsequently called mad cow disease, are many and quite varied. They include a loss of condition, unsteady gait, and a nervous or worried facial expression. Indeed, many vets regard the phrase mad cow disease as a misnomer. A much better name is worried or upset cow disease. And that is indeed very well illustrated in this slide here. You can see the expression on this animal who has BSE, lowered head, and a look of some concern, some puzzlement. Now, in November 1986, following this identification, the British government's Central Veterinary Research Laboratory in Weybridge, Surrey, announced a formal diagnosis of the disease based on the autopsies and brains of infected cattle. Now, these brains showed holes and lesions characteristic of a class of diseases of the central nervous system called the spongiform encephalopathies, in essence, spongy brain diseases. And the new cattle disease was therefore called bovine spongiform encephalopathy, or BSE for short. Now, these events in 1985 and 1986 heralded the start of what was to become a very major epidemic in the UK, in which roughly 1 million cattle were, to date, have been infected by the etiological agent of this disease. Now, the history of events since 1986 are well known to many, I'm sure in the audience, and indeed are etched on the minds of many civil servants and indeed a few politicians. It really only became a crisis in the broader public's mind in March of 1996, when the minister in charge of the Department of Health at that time, Mr. Stephen Dorrell, made an announcement to Parliament in the House of Commons that an advisory committee to MAF and the Department of Health, called the Spongiform Encephalopathy Advisory Committee, or SEAC for short, had suggested that 10 new cases of a new variant of the rare but rather lethal, or very lethal, Creutzfeldt-Jakob disease in humans was exposure to the etiological agent of BSE, prior to when regulations were introduced to prevent any part of cattle with clinical signs and specified offal from all cattle entering the human food chain. And those two events occurred in 1988 and 1989. Now, this announcement triggered a major crisis in Europe, both in consumer confidence in beef and beef products, and in relations between the member states of the European Union. Indeed, I'm sure many of you remember some of the headlines in the newspapers. This is one from The Independent, where the Commissioner for Agriculture, Franz Fischler, regarded, or was heard to comment, or commented, that this indeed was one of Europe's biggest crises. And then later on, a headline following some work of the Oxford Epidemiological Group that BSE, in fact, in cattle had actually been eaten by millions. Well, I will turn later on to the scientific issues involved here. Now, in this talk on the origins, pattern and control of this epidemic, I really wish to consider this from a variety of angles, scientific angles, including the nature of the disease agent, the factors determining its growth and decay of the epidemic, and most importantly, the impact of recent events, culling, and what the future holds in the shorter and longer term. Now, I'm going to start with the actual nature of the disease agent itself, and a little bit of history about the transmissible spongiform encephalopathies, TSEs for short. Now, if we look back through the historical literature, the first records of these neurological diseases date back to the early 18th century, with the mention of disorders in sheep, particularly in France, but also in Germany. In France, this disease was called trembling disease, tremblant, and in Germany it was called itching disease, or trotting disease, and these diseases today are known to be scrapie, which is a neurological disease of sheep, a fatal one. The different descriptions very much reflect the characteristics, clinical symptoms of these diseases, in other words, a poor condition, itching, trembling, staggering, and so forth. Now, for many years, from the early part of the 18th century, very little was understood about the disease, or this class of diseases, but particularly scrapie, first to be identified. And the concept that it was induced by a transmissible agent was not established until the middle of the 20th century, when two French scientists, Couillet and Chellet, reported transmission of scrapie by inoculation of healthy sheep by spinal material from contaminated or diseased sheep. And they noted a very, very long incubation period of between 14 to 22 months. And this indeed caused a lot of controversy in the scientific literature and community at the time, because nobody really believed that you could inoculate something and then have to wait for 14 to 22 months before the disease actually appeared. So it wasn't widely believed at the time. Now, subsequently, the disease, and this is perhaps the most important scientific point in this early history, was established in laboratory mice in the mid-1960s, and research continued, really, with much controversy still surrounding what was the nature of the transmissible agent. Now, with the volume of research on scrapie beginning to rise, here, for example, is a sheep infected with scrapie from a flock in Scotland. Again, very poor condition, rather confused stare in the eyes. Now, the volume of research really started to grow in the mid-1960s towards the 70s, but a very important event took place then. Scientists began to make an association between the clinical symptoms of scrapie in sheep and a disease kuru in the Fore-speaking people of the Eastern Highlands in Papua New Guinea. Now, these people were cannibals, and they had a particular custom, which was eating the brains of their deceased relatives in the belief that their knowledge would be passed on. And kuru is in fact the Force word for shaking or trembling. In other words, this was a very severe neurological disease. Now, it's not proved possible to determine when kuru first arose in Papua New Guinea, but anthropologists have certainly found evidence of the disease emerging in local consciousness and language in the early part of the 1900s. And the similarity between kuru, this disease in humans, and scrapie, the disease in sheep, with the destruction of the brain, the vacuolated spongiform appearance, was first noticed by an English scientist, Hadlow, in 1959, and within a very few years, a series of studies initiated by an American epidemiologist, Gadusink, in 1966, he reported the transmission of kuru by taking brain from deceased people to chimpanzees, demonstrating that this was a transmissible disease. And that work was to earn Gadusink in future years the Nobel Prize for Medicine. So as a consequence of this transmissible form, great interest in the scientific and medical communities about what this disease indeed was. Now, first of all, before turning into the nature, it is important to remember that kuru is one of a class of these diseases. Kuru is somewhat exotic. We're not cannibals in our own countries, so we're not transmitting on that form of the disease. But there's one in particular, Creutzfeldt-Jakob disease, which is a rare neurodegenerative disorder which causes this vacuolation and spongiform appearance of the brain and is lethal, which afflicts about 1 person per million head of population worldwide per annum. Very, very rare disease, but there are a set of these in humans. Now, one of the odd things about these diseases is in many cases there is an association with family. In other words, there's clear evidence of some sort of genetic inheritance. So you're more likely to get Creutzfeldt-Jakob disease in a sort of genetic linkage in family studies. It can also be transmitted, that has certainly been demonstrated, by contaminated equipment used in surgery, where the brain of a person or the spinal column is operated on in proper sterilization, which I'll turn to in a minute, of the equipment, and then transmission on to another patient. Now, the key scientific question with all of these spongiform neurodegenerative diseases, that's the ones in humans, scrapie in sheep, and BSE in cattle, is what on earth is the transmissible agent? And this really is a scientific puzzle of extraordinary interest. Now, clearly early on, we're talking about the mid-60s for the beginning of this, and technology was just developing scientifically to be able to look at viruses, the smallest of the pathogens. But of course, remember viruses and their larger counterparts like bacteria or protozoa or worms, all the pathogens, they all contain the genetic code which makes us function. In other words, DNA, our genetic code, the building block for the structure that we are. And clearly, one way to demonstrate transmissibility is to try to destroy that genetic code in the agent which is causing these neurodegenerative diseases. So a very simple way to do that is to inactivate by the usual methods that inactivate viruses and bacteria. And these could be very high temperatures. For example, temperatures in excess of 120 degrees C normally denature DNA. You could think of sterilization by formalin, intense high concentrations of formalin. You could think of intense slots of ionizing radiation. Or indeed, you could be more sophisticated. You could use proteases, in other words, enzymes, to try and chop up the DNA or the code. Now, none of these methods actually worked, which really did start to question whether this organism was a virus or indeed a bacteria. Now, an earlier suggestion that the infectious agent may be something totally different, in other words, a small protein, was made again by an English scientist, Griffiths, in 1967 in a short communication that was published in Nature. But it was not until, most people ignored that, thought it was quite a ludicrous suggestion, and the search continued for viruses and bacteria. And it was not until the early 1980s that the work of an American, Stanley Prusner, showed that infection was indeed associated with a smallish protein. And that protein is called the prion protein. There's a difference in American and English pronunciation, not an important one, prion or prion. And this protein is a very common one in cells in the central nervous system, the brain, spinal column, etc. And since that time, since Stanley Prusner's work, but remember, the precedent is Griffiths in 1967, the transmissible spongiform encephalopathies have often been called the prion diseases, protein diseases. Now here is just a shot of a cross-section of a mouse's brain infected with BSE to show this spongiform appearance. You see here these spongiform vacuoles which are created by this protein, and I'll explain in a minute how it does it. So this prion hypothesis, or prion hypothesis, of these transmissible spongiform encephalopathies has been seen by many, and this is the great scientific interest, as a challenge to one of the central dogma of 20th century biology, namely that the informational or coding molecule for replicating, in other words, living organisms, is indeed nucleic acid. So here we're saying, or suggesting, that a transmissible agent is a protein with no DNA, no RNA, and it can infect a host and replicate within that host. And that's an extraordinary concept. And I do stress that this idea is still controversial in the scientific community, but I again stress that the majority of specialists in this area are beginning to lean very heavily towards the prion-only hypothesis. Now, how on earth does it do it, and what actually happens? Now, that's a complicated question of which an entire lecture could be devoted to. So I'm going to give the very crudest of outline in the very simplest of terms. Most proteins, building blocks of our cells and our organs, bodies, and so forth, or all of them, have a genetic code, a gene which encodes for them. And the prion protein has a gene with a particular reading frame, and messenger RNA comes along in the cell, reads off this genetic code, and forms another template, which then goes into the cell's body, and on that template, amino acids are assembled to form the protein, the prion protein. And this is the normal form of that protein. And that in itself, numerous events can occur to formulate multiple molecules of this protein. Now, the event that creates an abnormal form, which is absolutely distinctive of the presence of disease, is not understood at present. If you put an abnormal form into a naive host that's making normal protein, then this abnormal form of the prion protein contacts the normal form, and when it does so, turns the normal form into the abnormal form. And of course, more normal forms are made, and it keeps on turning them into abnormal forms, and therefore it is replicating. Now, another event may occur where indeed de novo you may get abnormal production of a protein straight off the template. But how that occurs, again, is extremely poorly understood. What is the abnormal form? Well, it's nothing to do with the gene. The gene's the same, so it's a post-transcriptional event. It occurs after messenger RNA is formed, after the building blocks are put on the messenger RNA. It indeed is thought to be a structural change, a three-dimensional configurational change, where the normal protein has one three-dimensional configuration, the abnormal protein has a totally different one. But when that abnormal configuration contacts the normal one, it turns it into the abnormal one and replicates and multiplies. Now here, for example, is a slide again of a mouse brain, and these are plaques of abnormal prion protein, which are replicated and converting normal into abnormal, and beginning to start the process of causing this vacuolation and the destruction of the brain, the normal function of the brain. And I stress there's an awful lot of biochemistry and basic biology to be done to understand this disease process. And it's only by understanding it that you're usually led towards possible interventions, whether drugs, immunotherapy, or whatever. One of the crucial experiments in this area actually was performed by a Swiss scientist, Charles Weissmann, at Zurich, where he actually made what are called knockout mice, where you knock the prion gene out of the genome of the mouse by various techniques, and then you can show, or Charles Weissmann showed, that in fact that mouse with no prion gene cannot get diseased. So the presence of that normal gene is absolutely crucial to the development of abnormal disease. Before passing particularly to BSE, I'd want to stress that these diseases are somewhat more complicated than I've given a brief hint of, because in essence all sorts of patterns go on inside a host. And here's one that I'm going to return to later because it relates to risk to humans. If you take a mouse and infect it with a scrapie agent at time 0, the agent, this is the abnormal protein, replicates in different manners in different organs. This is diagrammatic, an example of in the spleen of the mouse and in the brain of the mouse. And this is a logarithmic scale here, so these densities are extremely high. Clinical symptoms appear about here, neurological disease, and eventually death occurs at a very high abnormal prion density. So this exponential rise in the abnormal form in the brain is associated with disease. If you think of transmissibility of an infectious agent, which is this abnormal form of the protein, it's highly probable, although not certain, that infectiousness is higher at the tail end of this long period than at the early period. But do notice this abnormal form is in other tissues, and it's currently believed that it travels around the body in part via the immune system, and therefore a variety of other tissues appear unsafe in terms of consumption. And that includes the spleen, that includes lymph nodes, that includes, of course, anything associated with the immune system. So that's one thing to bear in mind when I turn later on to the epidemic. The second thing is that, like most infectious agents, if this truly is such a thing, genetic background of the host is very, very important, and that influences us all. Some people, when they contract a cold, a virus, have very severe symptoms, others have very mild symptoms. And the mouse genetic background for a model by Bruce, mouse and BSE here, taken into mice, the type of genetic background of the mouse determines the incubation period, which is the time from when you infect the mouse to when it gets serious neurological disease. Similarly, and this is a great surprise, that I'm not surprised about, but if you passage this agent through different strains of mice, you can create different strains of the agent. Now what that is, I haven't got a clue, but it may be different three-dimensional configurations in the abnormal protein. And therefore you've got a complex background of different strains, different host genetic background, determining different courses of clinical symptoms. And although not certain as yet, it's probable that genetic background is very important in the BSE issue. Now, that is by way of an introduction, and I really now would like to turn very much to the epidemiology of BSE. And the first thing I want to comment upon is how dangerous is this organism? Can it be transmitted to other animals? Well, to date, you may be surprised to know that this beast, this abnormal protein, can be transmitted, and that's isolated in cattle, to deer, cats, dogs, sheep, goats, mice, marmosets, pigs, macaque monkeys. Probably, with our perspective, two of the most important are the primates, marmosets and macaque monkeys. So really, at a very early stage in this BSE epidemic, alarm bells should have been ringing, because this organism, or whatever it is, not an organism, this abnormal protein, can cross species barriers, and that's somewhat unusual for infectious agents. Now, my own involvement in this problem started in 1989 to 1990. I was a member at the time of the Advisory Council on Science and Technology, which advised ministers and the Prime Minister at the time on science policy issues. And therefore I had very ready access to the chief scientist of the day, who was Sir John Fairclough. I was concerned that the predictions about the rapid demise of the BSE epidemic made in 1989, the predictions that there was only one route of transmission, which was via infected cattle being turned into meat and bone meal feed and then fed back to cattle, that's contaminated feed, were indeed somewhat optimistic. And I talked to the chief scientist, John Fairclough, and at that time the chief scientist post changed over to Sir William Stewart, and also discussed it with Bill Stewart. And a letter was written suggesting that the Ministry of Agriculture, Fisheries and Food might like to get some external scientific advice from the medical epidemiological community to have an input into looking at the BSE epidemic. A very polite note was received back from MAF saying that they felt they had all the internal scientific expertise they needed at that time and indeed there's no necessity to bring in external input. Now, although this is with hindsight, it is a great pity, because if the techniques I was thinking about had been applied, and others have these techniques, then we would have known in 1990 that the feed ban, the stopping or trying to stop contaminated feed material going back into the ruminant food chain, wasn't working. In other words, contaminated feed was still going through the system. Well, this is water under the bridge now, and one really should turn to modern time. Now, what this is going to show is the slow growth of the epidemic spatially in the UK by quarter year. And you can see instantly from its origins down in the south, it spread right throughout the United Kingdom. And these red spots are the hot spots. Showing it again, spread very quickly throughout the United Kingdom. That's year and quarter of the year. The red spots, south of England, Kent over here, Norfolk and Kent. And you can see it's very rapid emergence and spread. And just a third time, showing its spatial evolution throughout the United Kingdom. Now, if we turn more specifically to the detailed history that's going to be important in what I'm going to say. 86, I've already mentioned that we have the disease was recognized, but the most important thing was a disease surveillance system was initiated by scientists at the Central Veterinary Laboratory, particularly under John Wiles Smith, a veterinarian. And that disease surveillance system was of high quality and is very important in what one can understand today. So very great credit to them. 1988, Sir Richard Southwood was asked by government to set up a working party to assess the health risks of BSE. BSE was made a notifiable disease very, very quickly. In other words, all vets who identified a case had to notify it to government. A ban on ruminant material in ruminant feeds was introduced in 1988, and indeed suspect and confirmed cases had to be slaughtered, prevent them from entering the human food chain. Southwood Committee reported back to government in 1989 and specified a bovine offal ban was in place. In other words, there was concern over material which might have contact with the immune system, indeed the gut as well, from entering the human food chain. And that was first for the human food chain, and then that was extended late in that year to all animals and birds. So clearly, government acted quickly on the results or the recommendations of the Southwood report. One thing they didn't react quickly to, which was a pity at the time, was to pay farmers full compensation, market price, for animals that were found to have BSE and had to be destroyed. And it took some time before an agreement of full compensation was put in place. And that meant, before 1990, there was underreporting of cases of BSE. Great pity. We turn now to a more recent time, 1996. This is where events started to move very quickly. SEAC, the Government Advisory Committee, reports possible association between this new variant CJD in humans and BSE, the BSE epidemic, with the implication that could have been a cross-species transmission event. In April, the European Community put a ban on all beef and all beef products emanating from the UK. And in June, John Major went to Florence to hammer out the Florence Agreement on a selective cull of cattle to relieve the ban on beef and beef products. And as we all know today, that was an unsuccessful attempt. But I should say that this design of this selective cull, the so-called Florence Agreement for a cull of about 140,000, 150,000 animals, was in no way based on scientific analysis, neither from the European Union's point of view or indeed from the British government's point of view. It was the construction of a political compromise. That may sound strong, but I feel strongly about it. And then in August, the full analysis of the epidemic and the impact of different culling policies. What happened here over this time was, for the first time in May of 1996, due to political and other pressures, MAF agreed to release the entire database, and they released it to one group, my own in Oxford, and we put together a team to work on the analysis of the epidemic and the implications of it. And it took from 1990 to 1996 to get the public release of this data. In October, John Collins at St Mary's did some elegant experiments on mice and demonstrated that the new variant CJD in humans, from autopsy or pathology of patients, looked very much to be linked with BSE. It's still not certain, that issue, but there's a definite suspicion there. If we move forward in 1996, in November, suddenly the government decided to implement and very wisely so, a cull of all cattle over 30 months of age. And indeed, that cull that took place from November to the early part of this year removed about 1 million animals from the UK's cattle herd. In April, because the European community still had not lifted the ban on UK exports, the government in addition implemented the Florence Agreement and put the selective cull in place. As I will tell you later on, that selective cull had virtually zero impact, as easily predicted. And then in this year, in April, and I'm going to talk a little bit about this work, scientific evidence for maternal transmission of this agent from mothers to calves, either prior to birth or just post-birth, became apparent. And I'll talk in detail about that because this work is unpublished as yet. Now, origins of the BSE epidemic, how on earth did the thing arise? Well, this is a speculation. Evolutionary events are always speculation. The speculation is that the scrapie agent in sheep somehow got into ruminant food, in other words, in meat and bone meal supplements, and a cross-species transmission event occurred to cattle. Then, most importantly, there was a change in the rendering process in the early part of the 80s, reduction in the level of temperature treatment, and a reduction in use of hydrocarbon solvents in the actual rendering process. And that is believed to have been important, although it may not be. That's speculation again. And then, most importantly of all, as shown in that spatial development of the epidemic, infected animals were turned into meat and bone meal feed for protein supplements for cattle diets, both for milk production and beef production, and that contaminated feed was disseminated via the production companies unknowingly throughout the United Kingdom. And that caused this recycling or fueling of the epidemic. Now, to turn to the epidemic. To understand this, there are a lot of bits and pieces that one needs to analyze, interpret, do experiments upon. And in a talk of this nature, I clearly can't go through them all, so I'm going to select a few processes to illustrate the sort of thought that needs to go on, the sort of scientific work that needs to take place. If you're thinking about transmission, it can occur basically in this type of problem via three routes. 1, consumption of contaminated feed. 2, maternal transmission from mother to calf, either prior to birth or post-birth. And 3, most sinisterly of all, it could occur via infected animals spreading by cow-to-cow contact or contaminating pasture. 3 possible routes. Now, the first 2 I'm going to show, there's obvious evidence for ruminant feed. The second one I'm going to show new evidence for. The third one, there is no evidence to date that you can contaminate pasture or get cow-to-cow transmission. But scientifically, you cannot exclude it. Just because you don't have evidence for it doesn't mean to say it doesn't occur. So I put a caveat and a caution around that area. Now, the first thing you need to quantify in some sort of way, and this is a rather bizarre illustration of the point, is by age, how much protein supplement do cattle consume, whether in dairy or beef herds? And this is some data from an experimental farm. And indeed, prior to our starting work on this, this sort of data had not been collated and been put together. But it just shows for a particular animal, this is one trajectory, it shows how daily protein intake, supplemented, possibly contaminated feed, changed through the age of the animal. So in other words, counter to popular perception at the time, protein supplements were not just used for young animals, young calves. They were indeed widely used throughout Europe in the agricultural industry to boost beef production and to boost dairy milk yield by supplementing feeds throughout all age classes. So therefore, all age classes of animals could be exposed to this agent. Now, given that information, you can use various statistical and other techniques to estimate the exposure, age-dependent exposure, which is 2 processes: amount of contaminated feed consumed and age-dependent susceptibility to the agent. And one knows from experimental work on mice and other models that indeed there is some age-dependent susceptibility. Younger animals more susceptible than older ones. And this is our estimate of the mean age of exposure/stroke susceptibility, about 1.3 years for cattle, but there's a variance and a long tail. Older animals can be infected. Of course, on a finer time scale, you can look at exposure across age classes and through time. And I just show this by way of passing because it shows, in essence, the time and season-dependent exposure, the estimated feed risk, the risk of infectious material being in meat and bone meal for cattle feed at quite a high resolution, and you can pick out the seasonality in this process. The seasonality is caused by an obvious thing. Farmers tend to supplement feeds with protein supplements more in winter months when grazing is poor, and that's reflected in this data. But most interestingly from this graph is this point. The ban on ruminant feed material entering the ruminant food chain was put in place in 88-89, and yet you can see that feed risk continued thereafter. In other words, knowingly or unknowingly, people were cheating in this system. So it's a great pity a detailed analysis of the epidemic hadn't taken place about that point in time. Now, if you look at the data itself, very ably collected by math scientists at the Weybridge Laboratory, then there's a lot of detail in it, and I'm only going to show a little bit of the detail. If you take given cohorts of cattle, and these are all dairy cattle, I'm, for the purpose of simplicity, concentrate on the dairy herd, 1989 born cohort, coming back to 1977 cohort, this cohort started to get exposed to this agent about 10 to 11 years after birth. So this thing was beginning to become apparent in the early 1980s, mid-1980s, and indeed this graph shows very clearly that older animals can be infected. Now, coming forward as the epidemic developed with this recycling and spread of the feed, then the average age of infection gets younger and younger. That's a well-known epidemic process. Now, that is the fraction of cases in different age classes. Clearly, one could do this in a variety of different ways, but one is to look at the total number of cases. The epidemic is developing over this time, and in nearer time, indeed, there are a very, very large number of cases by age. With the average age of exposure about 1.3, you're getting the mean age of infection somewhere about 5 to 6. So a lot of epidemiological detail in the database. Now, one of the most important experiments was done by Dr. Wells at the Central Veterinary Laboratory, which was to take cattle, unexposed cattle, feed them brain material from diseased animals, and follow them over time to assess the incubation period of the disease. In other words, time from exposure to when the disease developed. And he did these experiments with a variety of exposure levels, 1 gram through to 3 times 100 grams, very small quantity of brain tissue, and yet these animals still developed BSE on a time scale of about 50 to 60 months. So the incubation period is a little bit quantity exposure dependent, but it's a long incubation period, and indeed you can analyze the data in total and get an estimate that the mean incubation period from contamination, infection, to when BSE develops is 5 years with a moderate variance. So nothing is certain, there are always probability elements, so it's a probability distribution of the incubation period. Very long incubation period. Now, probably more important than this is the routes of transmission. And I just very briefly want to talk about some new research which is in press at the moment, and that is the issue of maternal transmission. That is enhanced risk in offspring born to mother who subsequently develops BSE. And that could be due to true transmission of an agent. It could be due to genetic predisposition, because the calf shares half of the genes of the mother. Or it could be due to some combination of both, genetic or maternal. Of course, this issue has been of some concern to the public. A cartoon by Matt just illustrates this. Clearly, it would be nice when you go to the butcher to know a little bit about the origins of the meat. And right at this moment in time, this issue of maternal transmission is causing some concern in the European Union, because how does the transmission occur if it does occur? Let me just review the scientific results first. 2 studies were conducted, one by scientists at MAF, the Ministry of Agriculture, a cohort study following calves born to mothers who developed BSE, and another study done by my own research group at Oxford, which was tracing the fate of all calves born to mothers who subsequently developed BSE. And then, essentially, the Oxford group and a group at Cambridge and Reading analysed both of these studies to come to a consensus about what the data meant, and that is the work that's in press at the present. Now here first is the tracing study. A relative risk of 1 is the borderline. In other words, anything below that or around 1 means there's no enhanced risk to calves born to infected or diseased mothers. A risk above 1 means there is some evidence of an enhanced maternal risk. And most interestingly, this risk is linked to the close period before mother goes down with BSE. In other words, if the calf is born close to when the mother is diagnosed with BSE, in this period here of about 18 months to just after onset, then that calf has a high risk of developing BSE. And that is a classical imprint of an infectious agent and undoubtedly is related to that exponentially rising prion density. So that's strong evidence for maternally enhanced risk, hint of strong evidence for an etiological agent. In other words, not genetic predisposition, true maternal transmission. The cohort study, the experimental study, was unfortunately flawed in design. And we didn't know this at the time of analysing the data, but the government scientists, and no fault of their own, because this is a very obvious thing to assume, they took calves from BSE plus mothers and calves from BSE minus mothers and followed them over time, but unfortunately they fed both groups meat and bone meal supplement. And because the meat and bone meal ban hadn't worked in 1989, both groups of calves were exposed to the infectious agent. But with a little bit of statistical jiggery-pokery, it's possible to analyse that data even with that bias. And the important columns are these two, and you can see calves born to BSE-infected mothers have a greater risk of themselves going down with BSE, and that risk is about 10% over 5 to 6 months. More importantly, if you look where that risk lies, you find yet again it lies close to birth. If the animal is born close to the period when the mother goes down with BSE, then the calf is more likely to go down with BSE. Strong hint of a transmissible agent. So in other words, this new data says there is a maternally enhanced risk. It strongly hints at an infectious agent being involved, but it cannot eliminate the possibility that genetic predisposition lies against this background and is an important component. So therefore, one cannot exclude both acting concomitantly. Now the implications of that, how does this agent get transmitted maternally? Is it via milk? Is it before birth? Is it after birth? That is totally unknown at present. Now, demography is pretty important too for cattle. The British cattle herd was going down in size due to EU regulations and quotas and so forth, and was down to about 10 million in 1994. But the most important thing about cattle, and this was something that surprised us very much in the research group when we were working on this, was the life expectancy issue. We'd been told by many people that once an animal went into a dairy herd, it had a very long life expectancy. Farmers tended to keep good quality dairy animals for 5, 10, so on years. Well, indeed, that is not the case. If you look at survival from birth, then the life expectancy is about 2 to 3 years. So in other words, farmers weed out the unproductive animals, send them off for beef production. Now this life expectancy profile is very, very important because just think, we've got an average incubation period of 5 years, we've got a life expectancy of the animal of 2.5 years. That means most infected animals do not develop symptoms of disease before they're slaughtered, before they enter the food chain. And at present scientific point in time, we have no diagnostic method for identifying infected animals except by the clinical symptoms of disease. So there's a 5-year period where you don't know the beast has got this abnormal protein, and many animals at a 2.5 year are going into the food chain, so prior to the control of contaminated feed or contaminated cattle in 1989 going into the food chain. So this demography is very, very important. Now, the epidemic itself was a classic bell-shaped form with a peak in 1992, and I've shown it here in years. There's a lot of other detail, as I've suggested, underlying it. Now, these are the cases of BSE because that's all you can record. So quite a critical scientific question is, how many infections lie behind those observed cases? Now, to solve that problem, you can use a particular set of techniques which I've tried to explain conceptually here. You have the epidemic, you know the incubation period from infection to diagnosis of disease. Knowing that probability distribution and that, you can use that to calculate when these animals with BSE got infected. So you back calculate the instance of infection, and then you look at more recent time points, and you can extrapolate off them forward into the future. You can't do it too far forward, but you can do it for a few years, perhaps even a decade. So that back calculation technology is available. It involves some quite sophisticated mathematics and statistics, and I'm not going to explain the detail of this, but just to say you have observations like the probability that a case gets reported, there's underreporting. You have the probability that a cow survives to an age, age-related exposure, probability that a cow is maternally infected, the incubation period distribution of the disease, and the feed risk function. And putting that all together and using some iterative numerical procedures, one can actually use this methodology to calculate the back trajectory of this epidemic and therefore the human risk, what we were exposed to. Now, to give you the results of such back prediction, they're very simple to interpret. This is what we've seen, the purpley bluey bit. Those are the cases of BSE that have been reported, but when you use these technologies, the red is the new infections per year. In other words, what we saw was the tip of an iceberg. Vast numbers of cattle were infected. Indeed, the numbers are here, about 800,000 infected before there were controls on this material going into the human food chain, and after 1989, when the controls were in place, a relatively small number of animals. So a small number of cases observed, but a huge number of infected animals in the system. And that's that problem of incubation period life expectancy. Now, you can check the results, of course. You can look at things like how well do these predictions fit past trends, and these are just to show you that the mathematics and the model, the fitted, is a very good descriptor of the reported instance of disease. So one always does a sort of validity check, a scientific validity check. You can look at patterns of how the age distribution, or by year of incubation of the disease, has changed over time. In other words, these are animals in their first year of incubating BSE. These are animals in their late years. And you can see that in the early phase of the epidemic, all animals were in the early stage of incubation. Late stages of the epidemic, more recently, in the more dangerous stages, when the abnormal prion density is high. You can represent that as numbers, and just remember it's this late stage of the incubation period, when abnormal prion densities are high, that is potentially the most dangerous. So here's the total number of animals in a slightly different way, looking at years before clinical onset, and you can see those very close to development of BSE. The larger numbers are here coming forward into the more recent times. The future is not too bad. This epidemic is in a phase of very rapid decay, and the red is the new infections, and the blue are the cases. And many of these are going to be maternally transmitted infections, and you can see they're falling to extremely low levels, and indeed there are wide confidence bounds on these, so a cautionary tale there, as in all prediction. But the epidemic is beginning to get to extremely low levels. Now, I want to just spend a few minutes on this because you have a lot of detail in the forward prediction about the year coming forward to 1999, the age of the animal, and the reported number of infections by age. Now, remember that the government in November decided to introduce the over the 30-month year cull. All animals over 30 months of age removed from the food chain in 1996, beginning the cull. So clearly, most of the cases were going to be in older animals in the future, so that over the 30-month cull has had an extraordinary influence in reducing the risk to us. Indeed, the European Union was so fussed with this selective cull of 140 and 150,000 animals that it really, and this is part the British government's fault, it should have stressed to the European Union to look at the scientific evidence and see that this 30-month cull was much, much more important. The Florence Agreement was an irrelevance. Coming into culling, you've got a whole variety of things that you can do. You can maximize the number of cases prevented. You can end the epidemic as quickly as possible. You can end the epidemic by a given year and so forth. You can use even economic constraints. You can take expenditure into account. And the only point I wish to make here is the scientific methods are there to precisely do each of these calculations. And yet never in the stages of the epidemic did either the European Union or the British government ask for those scientific calculations to help them in policy formulation. And that really was a missed opportunity. Now, just in a sense, you can do more subtle things. You can target herds, age groups. You can target animals born to mothers who went down with BSE, whole range of options. And to cut a very long story short, herd targeting by instance, combined with tracing animals born to infected mothers, is undoubtedly, in our view, scientific view, the best option. It maximizes the number of cases prevented and minimizes the number of animals culled. There's always in these problems a law of diminishing returns. As you cull more and more cattle, then the return, this is a logarithmic scale here, the return you get is less and less in terms of taking cases out of the future epidemic. So there are some very classical economic problems here, and indeed high expenditure is required to get a very high number of cases. Now I suppose the question that many of you would really like me to address is, what is the risk to humans? What is the likely pattern of this new variant CJD epidemic, and so forth? Well, unfortunately at present, it's not possible to be precise about this for a variety of reasons. First of all, the experiments of Professor Collins using mice, using the new variant CJD, transmitting it to mice, looking at the pattern of damage in the brain, was indicative of association between BSE and the new variant CJD in humans. But it wasn't proof, and there are more experiments to do there. Secondly, the number of new variant cases of CJD we've seen is somewhat small at present. Now, thirdly, over the 30-month cull is taking this epidemic down at a very rapid rate, and we really do expect the epidemic to be at almost negligible levels by year 2000. So I stress the point that the safety now is not an issue. It's what took place in the past, that huge number of infected animals that went into our food chain prior to 1989. So the future, one is really thinking of the decay, the rapid decay of this epidemic. This is what has been observed with the new variant CJD. This is the background reported annual cases, which are nothing to do with the new variant ones. They're the typical background of this neurodegenerative disease, a very, very rare disorder. So that's been going on for a long time. The new variant cases, I'm sorry this is a little bit indistinct, but you've got here in red the new variant cases reported. And we're only talking about a time span of 18 months to 2 years. And the total number of cases absolutely confirmed at autopsy at present is 16. There are many more lying behind that are suspect, but a significant number of those will not be confirmed. Now, many of you will have read in the press and the scientific literature projections about the future of this new variant CJD epidemic. Surely it's foolish, absolutely scientifically foolish, to say anything at this point in time. On the basis of 16 cases, seen over a period of 18 months to 2 years, the 1997 figure is totally incomplete as yet. You cannot say anything about the future. So here is an area that I really feel scientists should keep their mouths shut and say, I don't know. So that's a point I really want to stress with you. Now, to end this lecture, I would like to return a little bit to policy issues. And I'd like to draw your attention to 4 major conclusions, or indeed messages that I'd like to leave with you. The first one really does lie on this last point I've made, and that is that clearly you have to be extremely cautious. You shouldn't be somebody who is predicting doom on the basis of such a small amount of scientific information. All you can do is put in place the appropriate surveillance mechanisms and encourage research on a precautionary basis. If this is serious, can we stimulate basic biomedical research to try to think of ways of actually knocking out this abnormal protein? So the future, the point I want to leave with you is the future is very, very uncertain, even though we understand a lot about the past and the future of the BSE epidemic. It's the human bit that we understand very little. Second point I want to leave with you is we must revise our rules in agriculture about the use of animal feeds that contain animal material. Not just because of these TSEs, they are a sort of very serious story in themselves, but there are all sorts of other things. Viruses, bacteria, antibiotic-resistant bacteria, plasmid transfers. We've got to think very, very carefully about what we do or put in animal feeds. The third point I want to make, and this is somewhat more sensitive area, government departments must be open with information, not just to the scientific community, but to the public. As cuts in government department research budgets have taken an increasing toll over the past decades, government research institutes do not have the scientific expertise to tackle these new problems on the scale that is required. So they must be open to bring in external scientific advice as quickly as possible. That is, of course, a very directed comment at one particular ministry, and indeed other ministries are better in some senses, but not all are ideal in this area. Now, the fourth area, which is very much directed at my own community and myself as well, is that scientists should learn to say, I don't know the answer. It takes more courage to say, I don't know what the risk to humans is, than it does to give a somewhat firm opinion. And similarly, politicians must be prepared to repeat this statement, and where human health and safety is concerned, to adopt a precautionary principle. Now, you may think it's easy to say that with hindsight, when the future of an entire agricultural industry was at risk. But if you look at the scientific evidence of cross-species transmission of these TSEs, there was clearly a hint of danger there. And I would argue that the precautionary principle should have been applied with vigor. But I do certainly accept that it's easy to say that with hindsight, much more difficult to say it at the time. To end very firmly, I must indeed acknowledge a group of people. My own understanding of this problem has been enormously enhanced by a group of very talented young scientists who I've had the pleasure of working with at Oxford. So indeed a very warm thank you to them. And lastly, you may be wondering what this little squeezy object is. Well, it's a squeezy cow, and I'm told that it's very useful in relieving tension by squeezing it. And personally, this has been of enormous value in this BSE crisis over the past year in particular, but indeed earlier. Thank you very much indeed for your attention.